Imagine a runner sprinting at top speed toward a towering, completely solid brick wall.
Right.
According to the classical laws of physics, the rules of the macroscopic world we navigate every day, to get to the other side of that obstacle, the runner has to expend a massive amount of energy to climb over it. There's just no other way.
Exactly. You need to put the work in to get over the barrier.
Right. But instead of slowing down to climb, this runner accelerates. They hit the wall, and at the exact moment of impact, they don't crash. They just, phase right through the solid clicks.
Which is impossible.
Completely impossible. They emerge on the other side totally unscathed, like a ghost. Now, if we saw this happen in our everyday lives, we would call it magic, or, I don't know, a glitch in the simulation.
Yeah, you'd think you were losing your mind.
But right now, deep inside your body, subatomic particles are performing this exact impossible ghost trick millions of times a second. It's a scientifically verified quantum phenomenon called tunneling.
It really is, and it's happening constantly.
And what we are going to explore today is the totally mind-bending reality that without this ghost trick, your DNA wouldn't mutate, you wouldn't evolve, and honestly, you almost certainly would not be alive right now.
It completely upends our fundamental understanding of reality. Because these spooky, seemingly impossible rules of the quantum realm, things like tunneling, or particles gifting at multiple places at once, or sharing information instantaneously across vast distances.
Yeah, entanglement.
Right, entanglement. These have long been viewed as just quirky oddities. We really thought they were strictly confined to sterile physics laboratories, observable only inside vacuum chambers cooled down to absolute zero.
Because the real world is too messy for them.
Exactly. But the reality is far stranger. We are now discovering that these bizarre quantum mechanics are the actual engine driving life, evolution, and potentially even the deepest mysteries of the mind.
Well, welcome to the deep dive. We are taking a massive stack of incredibly dense research papers, historical lectures, and cutting-edge biological studies, and we are plunging straight into the field of quantum biology.
It's a fascinating area.
It really is. This is all about understanding the bleeding edge of how your body actually works at the most fundamental level. But to grasp how life operates, we kind of have to start with a deceptively simple question, one that was posed back in 1943 by the Nobel Prize-winning physicist Erwin Schrödinger.
Right, his famous Dublin lectures.
Yeah, he gave this lecture series, which later became a highly influential book, simply titled, What is Life? And to frame this massive question, Schrödinger actually looked inward. He made this deeply personal observation about his own family. He realized that his nose was perfectly identical to his grandfather's nose.
Which, sounds like a very basic observation of genetics. People inherit physical traits, obviously.
Sure.
But Schrödinger approached this not as a biologist looking at traits, but as a physicist looking at the fundamental building blocks of matter. He was thinking about atoms.
And atoms are incredibly chaotic. They are constantly churning, vibrating with heat energy, reacting, replacing themselves.
They never sit still.
Never. So Schrödinger is looking in the mirror and wondering how it is physically possible that the molecular arrangement forming his grandfather's nose, a structure formed a century ago in a warm, messy, 37 degrees Celsius human body, could hold its shape so perfectly across multiple generations.
It's a profound paradox when you really think about it.
Right. How does a living, breathing organism maintain such incredibly precise architectural order over decades without just collapsing into a puddle of molecular chaos?
What's really fascinating here is that Schrödinger was identifying a massive contradiction between the observable reality of biology and the established laws of physics. Specifically, he was wrestling with the second law of thermodynamics.
The big one.
The big one. This law is generally considered one of the most unbreakable rules of the universe. It dictates that any isolated system will naturally and inevitably degrade into a state of disorder. And this measure of disorder is what physicists call entropy.
Okay, I always think of this in terms of a sandcastle on a beach. Like, if you spend hours building a perfectly ordered, highly structured sandcastle, and then you just leave it alone, what happens?
It falls apart.
Exactly. The wind, the rain, the ocean waves, the natural chaotic forces of the environment, they will eventually break it down until it is just a flat, disordered pile of sand.
Because the universe constantly drives everything toward equilibrium, toward maximum chaos.
Right. But a living thing is essentially a biological sandcastle that refuses to fall down. It survives the waves, and then it builds extra towers and maybe a moat. How does it do that without violating the laws of physics?
Well, Schrödinger's brilliant resolution to this paradox was to point out a really crucial detail about the nature of living things. We are not isolated or closed systems.
We don't exist in a vacuum.
Exactly. We aren't sealed off from the universe. Living things are open systems. So to avoid decaying into what he called atomic chaos, a living organism has to actively feed on order.
Feed on order.
Yes. He termed this negative entropy, or negentropy. A living system must constantly extract orderliness from its surrounding environment to offset its own internal decay.
So we eat, we drink water, we breathe oxygen,
Right.
and plants absorb highly ordered packets of sunlight.
Yeah, we take in highly concentrated, ordered energy. An apple, for example, is a wonderfully complex, ordered arrangement of molecules. We ingest that apple, break down its chemical bonds, extract the order to maintain the structure of our own bodies,
And we expel the disordered energy.
spot on. We expel it in the form of body heat and waste back into the environment. A living being is fundamentally an order-extracting machine. We stay perfectly organized internally by constantly pushing chaos outward.
Okay, let's unpack this, because that means our very existence is basically a thermodynamic transaction.
It is.
We pay our entropy tax to the universe by radiating heat, and in exchange, we get to keep our molecular sandcastle intact for another day. That makes intuitive sense to me.
It's an elegant way to look at life.
But looking through the sources, this thermodynamic definition of life has been taken even further in recent years. There's a physicist from MIT named Jeremy England who derived this mathematical framework, suggesting that life isn't just some lucky 1 in a trillion cosmic accident.
Right. His work is really making waves.
His math suggests life might actually be an inevitable consequence of physics.
Yes. England's work is a profound expansion of Schrödinger's initial insight. He introduced this concept called dissipative adaptation.
Dissipative adaptation.
His equations, which, by the way, are grounded in established, undisputed physics, model what happens when you have a random group of atoms and you subject them to a consistent external source of energy, like the sun continuously shining on the primordial oceans of the early Earth.
Just blasting it with energy.
Exactly. His math demonstrates that under the right conditions, those random atoms will naturally begin to restructure themselves into increasingly complex patterns, and they do this for one specific reason:
Which is?
to dissipate that incoming energy more efficiently.
If I'm following this, you're saying that the universe has a fundamental problem. It has these localized clumps of concentrated energy, and it desperately wants to disperse them to cool down.
That's the core drive of thermodynamics, yes.
And life, like a complex self-replicating cell, is just the universe's most efficient way of solving that problem.
In a strictly thermodynamic sense, that is exactly the implication. The universe dictates that concentrated energy must disperse. What England's math shows is that a complex biological structure, living cell, is unbelievably good at absorbing energy and dissipating it as heat into a surrounding bath, like an ocean.
Wow.
And if you want to know what the absolute best way to dissipate twice as much energy is,
You make a copy of yourself, you replicate.
yes. The drive to replicate, the very essence of biological survival, can be viewed fundamentally as a mechanism to maximize energy dissipation.
The analogy he uses in the sources is striking. He argues that under the right physical conditions, the emergence of a living, breathing cell from non-living chemical soup should be as scientifically unsurprising as a rock rolling downhill.
It's a great analogy.
Right. Because a rock rolls down a hill because gravity dictates it must seek a lower energy state. It has no choice. And life emerges because thermodynamics dictates that matter must find the most efficient way to disperse energy.
It kind of strips the magic away from the origin of life, doesn't it?
It does, but it replaces it with something almost more awe-inspiring. It means life is woven into the very fabric of the physical universe.
And if we connect this to the bigger picture, it significantly blurs the line between living biology and non-living phenomena.
Because biology isn't the only thing that forms patterns, right?
Certainly not. Think about a hurricane. The ocean absorbs massive amounts of thermal energy from the sun. The atmosphere needs a way to disperse that heat. So a highly structured, incredibly organized spinning vortex or hurricane emerges specifically to churn that thermal energy upward and dissipate it.
Or a snowflake forming complex geometric patterns to dissipate energy as water freezes.
Exactly.
Or sand dunes arranging themselves into intricate ripples to dissipate the kinetic energy of the wind.
Yes. The equations of fluid dynamics describe the hurricane, meteorology describes the snowflake, and biological equations describe a cell. But underneath their specific scientific disciplines, they are all what physicists call dissipative structures. The physics of being alive is, at the macro level, the physics of processing energy.
Okay, but this brings us to a massive glaring problem. We've just painted a picture of life as this macro-level thermodynamic engine churning through energy like a hurricane. But engines have gears.
They do.
If we want to understand how this biological engine actually works, we have to zoom way, way in. We have to look at the individual molecules and atoms inside the cell. And when we cross that microscopic threshold, classical physics, the physics of thermodynamics and rolling rocks, breaks down entirely.
We enter the quantum realm.
We enter the quantum realm. And based on traditional physics, finding delicate quantum mechanics operating inside a living, breathing creature is supposed to be completely impossible.
It was considered the ultimate paradox for decades. The orthodox view in physics has always been that quantum states are exquisitely fragile.
Extremely fragile.
Phenomena like superposition, where a particle exists in multiple distinct states or locations simultaneously. Or entanglement, where particles are invisibly connected and instantly share information regardless of distance.
Right.
These are real phenomena. We know they exist, but they're incredibly sensitive.
Because to even observe these quantum effects, human physicists have to build massive, multi-million-dollar, hyper-isolated vacuum chambers. They have to cool the internal environment down to near absolute zero, colder than deep space, just to eliminate almost all thermal energy and vibration.
We go to all that trouble to prevent a process called quantum decoherence.
Decoherence.
Right. Decoherence is the sudden destruction of a delicate quantum state caused by interaction with the outside world. If a quantum particle in superposition gets bumped by a random bouncing molecule of air or vibrated by a tiny amount of heat, the quantum state collapses instantly.
Magic vanishes.
Exactly. The particle loses its special quantum properties and starts behaving like a normal, boring classical particle.
Now, contrast that perfectly silent, frozen, multi-million-dollar laboratory with the inside of a living human cell. A cell is famously described in biology textbooks as warm, wet, and noisy.
It's a very hostile environment for a quantum state.
It is quite literally a chaotic hurricane of thermal vibrations, bouncing water molecules, electrical gradients, and frantic chemical reactions.
Which is exactly why the idea of quantum biology was heavily dismissed for so long. The environment is simply too chaotic.
People just thought it was a romantic myth.
Pretty much. The sources highlight a pivotal moment in this debate. There's a calculation performed by the prominent physicist Max Tegmark in the year 2000. He wanted to settle the debate mathematically.
You wanted to put a number on it.
Right. He calculated how long a delicate quantum state could theoretically survive inside the warm, wet environment of the human brain before decoherence destroyed it.
And the number he came up with was 10 to the power of negative 13 seconds. For context, that is a decimal point, followed by 12 zeros and then a 1.
It is a fraction of a second so small it almost loses meaning.
It's unfathomably fast. The argument was that the quantum state is destroyed so quickly that biology couldn't possibly use it to perform any meaningful function.
Tegmark's calculation stood as a seemingly impenetrable mathematical wall against quantum biology for years. It appeared to definitively prove that biological systems were simply too hot and too chaotic to support anything beyond classical chemistry. But the magnificent twist that recent research has uncovered completely flips this narrative. Biology hasn't just found a magical, mysterious way to survive the noise of the cell. It has spent billions of years evolving to actively harness it.
This is where we need to look at the gears of the machine. Because to prove that life is actively using quantum tricks, the sources direct us to look at the absolute workhorses of the human body, the enzymes.
Enzymes are incredible. They are specialized proteins that act as biological catalysts. Their primary job is to accelerate chemical reactions inside your body.
And when we talk about acceleration in biology, we are talking about absurd, almost incomprehensible magnitudes, right?
Truly absurd. Enzymes speed up chemical reactions by a factor of 10 to the power of 12, up to 10 to the power of 17 times faster than those reactions would occur on their own in water.
Let's try to visualize that speed up. 10 to the power of 17. There are certain vital chemical bonds in our bodies that need to be broken constantly for our cells to generate energy and survive. If we didn't have enzymes to help, and we just waited for those molecules to naturally break apart at body temperature.
How long would we be waiting?
Yeah, how long?
For some specific reactions, the natural half-life without an enzyme is millions of years. For others, it approaches the entire age of the universe.
The age of the universe.
The chemical bonds are simply too stable to break on their own at 37 degrees Celsius. Yet with an enzyme present, That same reaction happens in milliseconds.
So an enzyme takes a process that should take 14 billion years and finishes it in the blink of an eye. How is that physically possible?
I was always taught the lock and key model in high school biology.
Most of us were, yeah. The idea that an enzyme is just a perfectly shaped mechanical mold. It grabs a specific molecule, physically bends it like a pair of pliers, stretches the chemical bond to its breaking point, and allows the reaction to happen.
The lock and key model was incredibly useful for a long time, but it is ultimately a classical macroscopic explanation. And as researchers got better at measuring the exact thermodynamics of these reactions, the math simply stopped adding up.
It wasn't enough.
No. Even if an enzyme perfectly mechanically bent a molecule, the energy barrier, the sheer amount of force required to snap that chemical bond, was still vastly higher than the thermal energy available inside a normal cell. Mechanical bending wasn't enough to explain the staggering speed.
It's the runner sprinting at the wall again. The classical physics says the runner doesn't have enough energy to climb over the wall, even with a ladder.
Precisely. And this is where quantum tunneling comes into play. We now have concrete evidence that enzymes are utilizing hydrogen and proton tunneling in vast numbers of essential biological chemical reactions. A single proton needs to be moved from one molecule to another to break or form a bond. Think of the energy required to rip that proton away as a towering physical mountain.
Okay, got the mountain.
Classical physics dictates that the enzyme must provide enough kinetic energy to push the proton all the way up and over the peak of the mountain. But quantum mechanics tells us that subatomic particles aren't just solid little spheres. They also exist as probability waves.
Waves of probability.
Yes. Because the proton is a wave, it doesn't have to go over the mountain. The wave function can simply extend through the barrier.
It phases right through the solid brick wall of the energy barrier. But wait, if we can't put a tiny camera inside a protein to film a proton phasing through a wall, how on earth do we actually know this is happening?
We prove it through a profoundly elegant piece of experimental science called the kinetic isotope effect, or KIA.
A kinetic isotope effect.
It relies on a very simple swap. A standard hydrogen atom is the lightest element in the universe. Its nucleus is made of just one single proton. But nature also provides heavier isotopes of hydrogen.
Right, they have extra neutrons.
Exactly. There is deuterium, which contains a proton and a neutron, making it roughly twice as heavy, and tritium, which has a proton and 2 neutrons, making it 3 times as heavy.
So the researchers basically trick the enzyme. They take the biological fuel the enzyme normally processes, but they swap out all the normal light hydrogen atoms with the heavier deuterium atoms.
Yes. Now, let's look at this through the lens of classical physics. If the enzyme is just a mechanical set of pliers, physically bending a molecule to break a bond, swapping a normal hydrogen for a slightly heavier deuterium, shouldn't matter that much.
Right, it's just a tiny bit heavier.
It might slow the reaction down by a tiny fraction because the molecule is slightly more massive, but the mechanical leverage is still exactly the same.
But quantum tunneling doesn't care about mechanical leverage. I know from reading the sources that the math governing how a wave function penetrates a barrier is highly dependent on mass.
It is extremely dependent on
The
mass.
heavier a particle gets, the dramatically harder it is for it to act like a wave and tunnel. The probability of it phasing through the wall shrinks exponentially.
It shrinks drastically. So researchers took specialized flavoenzymes, things like morphinone reductase and PTN reductase, and they fed them the heavier deuterium substrate. The results were undeniable.
What?
The reaction rates didn't just slow down a little bit, they absolutely plummeted.
Wow.
The slowdown was vastly larger than what classical mechanical physics could ever account for. The only mathematical model that accurately fits the massive drop in speed is that the normal, lighter hydrogen is successfully quantum tunneling through the energy barrier, and the heavier deuterium is struggling to wave penetrate the barrier because of its extra mass.
That is genuinely incredible. It means the old high school textbook model is fundamentally incomplete. An enzyme isn't just a mechanical pair of pliers, it is a highly evolved, dynamic quantum handshake.
A quantum handshake, yes.
The protein structure is literally facilitating a subatomic ghost trick. But that brings us back to the noise problem. How does the enzyme create the perfect, delicate conditions for a proton to tunnel when the cell is boiling with chaotic thermal energy?
Well, the sources detailed 2 distinct mechanisms evolved by proteins to handle this: passive dynamics and active or gated dynamics. Both of them, remarkably, rely entirely on the chaotic thermal noise of the cell.
Let's start with passive dynamics. Reading through the research, it sounds almost like the enzyme is just sitting there waiting for the universe to accidentally hand it a winning lottery ticket.
That is an apt comparison. In passive dynamics, the active site of the enzyme, the pocket where the chemical reaction takes place, is relatively rigid. The enzyme holds the target molecule in place, but the proton still can't tunnel yet.
It's stuck.
It's stuck. It has to wait for the natural chaotic thermal vibrations of the surrounding water molecules to jostle the enzyme. It gets violently bumped and vibrated millions of times a second.
Just getting beat up by the cell.
Pretty much. Eventually, purely by chance, one of those random chaotic vibrations momentarily forces the chemical bond into the exact perfect configuration. In that fleeting microsecond, the energy states of the two molecules become degenerate.
Degenerate meaning they perfectly match up.
Yes, they perfectly match up, and the proton tunnels across the gap.
So it's using the hurricane. It's letting the wind blow it around until, for 1/1000th of a second, is perfectly aligned. And the evidence for this passive strategy is that the tunneling rate in these specific enzymes is temperature independent.
Right.
Whether the surrounding fluid is a bit warmer or a bit cooler, the reaction rate stays the same. The enzyme is just patiently waiting for the chaotic dice to roll the right number.
Yes, the enzyme PTN reductase operates largely on this passive principle. But then there are enzymes that use active or gated dynamics. And this is a much more aggressive, highly choreographed quantum manipulation.
How does gated dynamics work?
In enzymes using gated dynamics, like morphinone reductase, the protein structure is not rigid at all. It is highly mobile and flexible. The protein doesn't just wait for a random vibration to align things perfectly. The protein structure itself acts like a specialized flexing accordion.
An accordion?
It absorbs thermal energy from the chaotic cellular environment and uses that kinetic energy to physically flex and slam the two molecules together. It actively compresses the physical distance between the proton donor and the acceptor.
So it's literally squeezing the energy barrier. If the barrier is a brick wall, the protein uses ambient heat to physically smash the wall thinner, so the proton has a significantly higher mathematical probability of tunneling through.
Exactly. Because this physical squeezing requires continuous kinetic energy to power the protein's movement, the tunneling rate in these active enzymes is highly temperature dependent.
Oh, that makes sense.
Right. The warmer the cell, the more kinetic energy the protein has to squeeze the barrier, and the faster the quantum tunneling occurs. We literally have evolved nanoscopic protein machines inside our bodies that exist solely to harvest chaotic heat and use it to manipulate the quantum wave functions of subatomic particles.
Here's where it gets really interesting.
Wow.
Because if quantum tunneling allows our enzymes to keep us alive on a millisecond by millisecond basis, it also allows biology to fundamentally change and adapt over millions of years. Tunneling isn't just powering the engine, it's rewriting the blueprints. And this brings us directly to DNA.
DNA replication is arguably the single most vital, consequential process in the history of biology. The continuation of life relies on extreme, almost unfathomable copying fidelity.
Let's put the scale of that fidelity into perspective for a second. Human DNA is made up of about 3 billion individual nucleotides, the basic letters of the genetic code, A, C, T, and G. The exact sequence of those 3 billion letters dictates how to build a human being. A tiny 0.1% variation in that massive sequence is all that separates your physical traits from mine. We are 99.9% identical.
It's astonishing when you think about it.
But if just one single letter gets copied incorrectly, out of those 3 billion, a single point mutation, it can cause devastating genetic disorders, sickle cell anemia, or trigger a cascade that leads to cancer. The biological machinery copying that code has to be flawless.
And yet, paradoxically, if that copying process was 100% perfect, life would have never evolved past single-celled organisms sitting in primordial mud.
We'd just be stuck.
Exactly. Mutations are the absolute driving force of evolution. Biology requires errors to explore new genetic designs. And the fundamental physical mechanism behind these crucial copying errors, the genesis of biological diversity, appears to be deeply tied to quantum mechanics.
To understand how a quantum error happens, we have to look at the physical structure of DNA. We all know the famous double helix shape. It looks like a twisted ladder. The long sides of the ladder are a strong sugar phosphate backbone, and the horizontal rungs of the ladder are the base pairs. The A is always connecting to the T's, and the G is always connecting to the C's. Now, what actually glues those two halves of the rung together in the middle?
Hydrogen bonds.
Hydrogen bonds,
Yeah.
which, as we established with the enzymes, are just single protons.
Yes. The two massive twisting strands of the DNA molecule are delicately zipped together down the middle by shared protons. Under normal, stable conditions, the proton sits closer to one side of the strand than the other.
It has a spot it likes.
Right. It rests in a stable, well-defined energetic valley on one half of the rung. But because these are protons, they're waves, and they're subject to the same quantum tunneling we saw in enzymes. A proton holding the DNA together can spontaneously tunnel through the energetic barrier separating the two strands and literally leap across the gap, lodging itself on the wrong side of the rung.
The ghost phases through the wall and ends up on the opposite strand of the DNA molecule.
This specific event creates what quantum chemists call a tautomeric form.
Tautomeric form.
It is a rare transient state. The chemical structure of the DNA base is technically slightly altered because a proton is physically in the wrong location. Now, in the vast majority of cases, the proton simply tunnels back a microsecond later and no harm is done. The DNA returns to its stable, classical state.
And this is the crux of the entire evolutionary process. What happens if the cell decides it is time to divide and the molecular machinery unzips the DNA ladder to make a copy at the exact fleeting millisecond that the proton is in that tunneled tautomeric state?
Then you have a permanent quantum typo.
Wow.
When the DNA helicase enzyme unzips the two strands, they separate. If the proton was caught on the wrong side during the split, the replication machinery moving along the strand reads the genetic code incorrectly.
Because it's literally reading the physical shape.
Exactly. The machinery identifies the genetic letters based on their physical shape and proton placement. If the proton has tunneled, the shape is subtly wrong. A guanine G might suddenly structurally mimic an adenine, an A, so the replication enzyme grabs the thymine, a T, to pair with it, instead of the cytosine, a C, that it was supposed to grab.
The newly synthesized strand of DNA is permanently physically altered. The genetic blueprint has been rewritten because a subatomic particle acted like a wave for a millionth of a second.
It is a staggering realization. It heavily implies that the sprawling, magnificent diversity of all biological life on Earth, from the simplest archaea to giant redwoods to the human brain, might fundamentally be driven by quantum errors.
The universe is exploring biological design space through the random tunneling of protons.
Yes, and what is truly remarkable is that in that same 1943 lecture we started with, Erwin Schrödinger presciently hypothesized this exact mechanism.
Wait, really? He guessed that?
He did. 10 years before Watson and Crick even modeled the double helix structure of DNA, Schrödinger argued that genetic mutations must be quantum leaps. He knew classical physics couldn't account for the sudden, discrete jumps in genetic traits. He deduced that life must be exploiting the quantum realm.
It is just wild to sit here and realize that the reason we aren't still just single-celled sludge is because protons occasionally decide to tunnel through energy barriers inside a warm cell.
It's humbling, honestly.
So we've firmly established that life uses quantum tunneling internally. It powers our enzymes and mutates our DNA. But what about the outside world? Do biological organisms use quantum mechanics to interact with their environment on a macro scale?
The sources emphatically say yes. Life uses quantum tricks to eat, and it uses them to navigate.
Let's begin with eating, which at the absolute base of the global food chain means photosynthesis. Plants, algae, and some cyanobacteria capture raw photons of sunlight and convert that electromagnetic energy into storable chemical energy.
It is, without exaggeration, the most important chemical reaction on the planet.
Because everything eats plants, or eats something that ate a plant. Every calorie of energy we burn ultimately traces back to a leaf sitting in the sun.
But the long-standing mystery of photosynthesis was its unbelievable physics-defying efficiency. When a photon from the sun strikes a light harvesting complex in a plant cell, it excites an electron, creating a little packet of energy called an exciton.
An exciton?
This exciton has a daunting task. It has to travel through a dense, chaotic forest of chlorophyll molecules to reach a specific destination called the reaction center, where the energy can actually be harvested and stored.
Now, if we model that exciton like a classical particle, like a pinball, it should just bounce around randomly from molecule to molecule, right? It hits a chlorophyll, bounces left, hits another, bounces right, desperately searching for the exit.
And that's how classical physics would describe it.
Classical physics dictates that every time it bounces, it loses a tiny bit of energy as heat. By the time a pinball randomly finds the reaction center in that maze, it should have lost a massive amount of its energy.
But it doesn't. Measurements show the exciton finds the reaction center with near-perfect 99.9% efficiency.
99.9%.
It loses almost no energy. Classical random walk thermodynamics simply cannot explain that level of perfection.
Because it's not acting like a pinball, what is it acting like?
The evidence strongly points to quantum coherence. The excitant doesn't bounce around randomly looking for the right path. Instead, the energy packet enters a state of quantum superposition. It acts as a wave, spreading out and exploring all possible paths through the chlorophyll maze simultaneously.
Okay, it's like if you had to drive across a massive, congested city during rush hour. A classical driver has to guess which route is fastest. They pick a road, get stuck in traffic, and lose time.
Right.
But a quantum driver could clone their car a million times, drive every single street and alleyway simultaneously, instantly determine which single route has zero traffic, and then immediately collapse all the clones into that one successful car, arriving perfectly on time.
That is an exceptionally good analogy. The energy exists in a coherent wave state, simultaneously feeling out the entire energetic landscape of the protein complex. Once the optimal fastest energetic pathway to the reaction center is identified by the wave, the superposition collapses and the energy is delivered almost instantaneously.
Wow.
Now, this brings us right back to the central paradox we discussed earlier with Max Tegmark. How does this incredibly fragile, coherent quantum wave survive in a green leaf baking in the hot sun without instantly decohering into random noise?
Right, because that leaf is warm and noisy.
Exactly. We will explain the specific mechanism of how the cell handles that noise in a moment. But first, we should look at the other major macro-scale quantum effect the sources highlight: avian magnetoreception.
I am absolutely obsessed with this topic. Birds migrating across the globe. We have known for a very long time that certain birds, like the European robin, can navigate across thousands of miles of featureless ocean by physically sensing the Earth's magnetic field. They have a biological compass.
They do.
But the Earth's magnetic field is incredibly frustratingly weak. It's significantly weaker than the cheap magnet holding a photo to your refrigerator. How can a warm, biological creature possibly detect a field that weak?
The answer lies in a specialized, highly complex protein called cryptochrome, which is located in the retina of the bird's eye.
In their eye?
Yes. The process begins when a single photon of blue light enters the bird's eye and strikes the cryptochrome protein. The energy from that photon knocks an electron out of its normal position, transferring it to a neighboring molecule within the protein. This creates what quantum chemists call a radical pair, two molecules, each now possessing an unpaired, highly reactive electron.
And because these two electrons were created together in the exact same event, they are entangled.
Yes, they share a quantum state. Even as they move slightly apart within the protein, their spin states, a fundamental quantum property akin to angular momentum, are intimately, invisibly correlated.
Spooky action at a distance.
Exactly. Now, the critical factor is that the spin states of these two electrons, whether they are spinning in parallel, meaning the same direction, or anti-parallel, opposite directions, strictly determine the chemical fate of the radical pair.
Okay, so it changes what happens next.
Right. Will the electrons simply fall back into their original places and recombine, ending the process? Or will the radical pair react further to create a distinct chemical product, which then generates a signal that the bird's optic nerve can read?
And this is where the weak magnetic field comes in.
Because these electrons are quantumly entangled, they are exquisitely, almost unbelievably sensitive to external magnetic fields. The incredibly weak magnetic field of the Earth is enough to gently tilt the probability of the spin states of these entangled electrons.
Oh, I see.
Depending on the physical angle of the bird's head relative to the Earth's magnetic field lines, the ratio of parallel to anti-parallel spins changes. This subtle quantum tilt alters the amount of chemical product produced by millions of cryptochrome proteins across the bird's retina.
So the bird isn't just feeling a magnetic pull like a compass needle in its brain. It is literally seeing the magnetic field.
Yes.
Is it like a filter over their vision? Like a pilot wearing a heads-up display in a fighter jet where they see the physical trees and mountains, but layered over that is a glowing chemical pattern indicating magnetic north, entirely generated by quantum entanglement in their eyeball.
That is the leading hypothesis among sensory biologists. It is a visual overlay dictated by quantum spin dynamics.
Hold on, I have to push back here, because when you describe a bird seeing quantum entanglement in its eyeball, it sounds like pure science fiction. Are we absolutely 100% sure this isn't just some complex, regular, classical chemistry we haven't fully mapped out yet?
It is vital to maintain that skepticism, but the constraints required to explain the experimental data simply cannot be met by classical chemistry. First, the sensitivity required to detect a field as weak as the Earth's using standard chemical binding energy is impossible. The thermal noise, the simple body heat of the bird, would completely drown out the tiny chemical signal. The radical pair mechanism is uniquely sensitive to weak fields, precisely because it relies on quantum spin, not chemical binding energy.
Okay.
Furthermore, the behavioral experiments are stunning. Researchers place these robins in specialized enclosures and expose them to extremely weak radio frequency electromagnetic fields.
Radio waves. But radio waves are completely harmless to biological chemistry. They don't have enough energy to break bonds.
Exactly. Classically, they should do nothing. But these specific radio frequencies perfectly match the resonance frequencies required to disrupt the delicate quantum spin states of entangled electrons.
Oh, wow.
When the radio frequency was turned on, the birds completely lost their ability to navigate. They fluttered around aimlessly. When the field was turned off, their internal compass instantly returned.
That's incredible.
The only theoretical framework in physics that accurately predicts all of these behaviors, the light dependency, the shear sensitivity, and the radio frequency disruption, is the quantum radical pair mechanism.
Okay, so we have enzymes using quantum tunneling to break bonds, DNA mutating via quantum proton typos, plants using superposition to perfectly route energy, and birds using entangled electrons to see magnetic fields. We have firmly, scientifically established that biology uses quantum mechanics.
Oh, yeah.
But we still haven't fully answered the most important question: how? How does the warm, wet, and noisy environment of the cell not instantly destroy these delicate quantum states? We mentioned earlier that biology somehow harnesses the noise. How does it do that?
This brings us to a radical new theoretical framework discussed in the most recent sources, one that completely reframes the fundamental relationship between biological life and quantum mechanics.
Reframes it how?
Well, the traditional view, the one that caused Tegmark and others to be so skeptical, always cast the living cell as a struggling, desperate engineer. It assumed the cell was trying to build a perfectly silent, isolated room in the middle of a hurricane to protect its fragile quantum computers.
Right. The assumption was that the cell is constantly fighting a losing battle against heat and vibration to keep the quantum wave alive for just one more microsecond.
But this new perspective argues that our entire premise was backward. The living cell doesn't fight the hurricane of cellular noise. The hurricane is the engine.
The hurricane is the engine.
The theory proposes that biological systems have evolved to be what are called decoherence engines.
Decoherence engines. That sounds like an oxymoron. Because as we discussed earlier, decoherence is usually the villain of the story. It's the thing that collapses the wave function and destroys quantum states.
In a quantum computer built by human engineers at Google or IBM, yes, decoherence is the ultimate enemy. We want our artificial qubits to remain in superposition for as long as possible so we can run complex algorithms in factor massive prime numbers.
Right.
But a biological organism doesn't care about factoring prime numbers. Life is trying to make highly specific, split-second functional decisions to survive.
Yeah.
The decoherence engine model proposes that biological systems have evolved to actively use and structure environmental noise to force a computation to conclude.
Let me see if I can picture this. Let's go back to an unfolded protein trying to figure out how to fold into its functional shape. It's not just sitting there as a classical string of amino acids guessing shapes one by one.
No.
It exists in a massive, blurry quantum superposition of billions of possible folded configurations simultaneously. The noise, the chaotic bouncing water molecules, the heat, the constant electrical vibrations of the cell acts like a chisel.
A chisel, I like that.
The noise interacts with the quantum blur. But the environment of the cell isn't totally random. It is highly structured by billions of years of evolution. The water molecules bounce in very specific, confined geometry, so the structured noise rapidly chisels away, or collapses, all the useless, unstable folding possibilities in the superposition, leaving only the one highly stable, functional biological outcome standing.
That is a phenomenal analogy. The cellular environment acts as a massive parallel selection mechanism. It is a process of controlled, targeted collapse not an endless random search.
It's sculpting it.
Yes, the protein doesn't classically search through a billion configurations over thousands of years. It explores them all instantaneously in superposition, and the highly structured noise of the cell rapidly decoheres all the non-functional pathways. The correct biological answer is simply the only shape robust enough to remain standing after the structured noise has collapsed everything else.
Yeah.
In this model, decoherence itself becomes the primary act of biological computation.
That is completely profound. It flips the entire paradigm of biology on its head. The noise, the heat, the chaos, it isn't a glitch in the system. It is the operating system itself.
And this connects beautifully back to the thermodynamics we discussed in the very beginning with Jeremy England.
Oh, how so?
To maintain this highly structured noise, to carefully manage this complex landscape of quantum information and force it to collapse correctly, the system requires continuous energy. The cell effectively pays a toll in classical entropy to collapse quantum uncertainty into functional classical certainty.
Everything has a price in physics.
Exactly. This paradigm gives us an entirely new, rigorous way to measure biological complexity, which the sources define as bioquantum algorithmic complexity, or BQAC.
Bioquantum algorithmic complexity. How is that different from how we normally measure how complex a living thing is? If I want to measure complexity now, I just count the number of genes, right? Or measure the size of the genome.
Classical metrics, like simply measuring the megabytes of data in a DNA sequence, only capture the static blueprint. They completely miss the quantum dimension of how that blueprint is actively processed.
I
The
see.
BQAC wouldn't just measure the static physical state of a cell, it would measure the computational process that actively generates and sustains it.
Yeah.
It defines complexity as the specific efficiency with which a biological system can manage a vast initial quantum state space and use environmental noise to collapse it into a functional result.
Okay, so a sliding scale.
Right. Under this metric, a virus has a relatively low BQAC, a bacterium is higher, and a human neuron processing sensory input would have an astronomically high BQAC.
That sounds like an incredibly elegant theoretical framework for biology. But as with all science, theory requires physical proof. How do we test if a living cell is actually operating as a decoherence engine? We can't just stick a regular glass thermometer in there and measure it.
This is where experimental physics gets truly fascinating. The sources detail a proposed experimental test utilizing something called a quantum biological probe, or QBP. This specifically involves the use of nitrogen vacancy centers in nanodiamonds.
Wait, nanodiamonds? We are proposing literally putting microscopic diamonds inside living cells.
Yes, a nanodiamond is exactly what it sounds like, an incredibly tiny crystal of diamond, often just a few nanometers across. Now, a perfect diamond is just a pristine, repeating lattice of carbon atoms. But we can engineer a specific defect into that lattice.
A purposeful defect.
Right. We remove two adjacent carbon atoms. In one of those empty spaces, we insert a single nitrogen atom. The space next to it is left completely empty of vacancy. This specific pairing is a nitrogen vacancy, or NV center.
Okay, we have a tiny diamond with a nitrogen atom sitting next to an empty hole. Why is that useful for biology?
Because that nitrogen atom has an extra electron that sits hovering in that empty vacancy. And because it is trapped inside the rigid diamond lattice, the quantum spin state of that specific electron is highly isolated, yet exquisitely, phenomenally sensitive to incredibly tiny changes in the local magnetic and electric fields surrounding the diamond.
It's like an antenna.
Exactly. We can read the spin state of that electron by shining a tiny laser on the diamond. The color of the light the diamond emits back changes depending on the electron's spin. It essentially acts as a highly advanced quantum stethoscope.
So the experimental idea is we attach one of these tiny diamond stethoscopes to a specific enzyme. As the enzyme works, as it enters its quantum superposition and then violently collapses into a functional state via the decoherence engine, its electrical shape changes, creating a microscopic magnetic ripple.
Yes, a ripple.
The electron in the diamond feels that ripple and changes its spin, and we can watch that happen in real time by looking at the light it emits.
Exactly. The quantum biological probe would allow us to literally listen to the real-time computational rhythm of decoherence happening inside a living molecule.
That is just so cool.
It is. If the decoherence engine theory is correct, the readout from the diamond wouldn't just be random, messy thermal static. We would detect a highly structured, rhythmic mathematical signature of the quantum state, repeatedly collapsing into classical function. It provides a concrete, empirical path to verify that biology is actively using noise to compute reality.
If that experiment works, it revolutionizes every single thing we thought we knew about biology. And naturally, that brings us to the ultimate, inescapable question. If the entire body, the tunneling enzymes, the mutating DNA, the sensory organs is operating as a massive synchronized quantum decoherence engine, what about the most complex structure in the known universe? What about the human brain? Does quantum biology explain consciousness?
This is the frontier where we must proceed with immense scientific caution and rigor.
Yeah, it gets a bit muddy here.
Very muddy. The intersection of quantum mechanics and human consciousness is a landscape heavily populated by media hype, pseudoscience, and our own deep philosophical desire to find a mystical explanation for our minds. However, there are highly rigorous, serious theories being explored by major physicists and neuroscientists.
The most famous theory, the one that always comes up in this debate, is ORCH-R, Orchestrated Objective Reduction. This was proposed by the Nobel laureate physicist Sir Roger Penrose and the anesthesiologist Stuart Hameroff.
A very famous collaboration.
Yeah. The core idea is that tiny structural tubes inside our neurons, called microtubules, are acting as organic quantum computers, and that the orchestrated collapse of their quantum states across the brain is what physically generates our conscious experience.
Yes. And recent discoveries in quantum biology have fundamentally shifted the burden of proof regarding ORCH-R. For decades, the primary overriding criticism of Penrose and Hameroff was the Tegmark calculation we discussed at the beginning, the assertion that the brain is simply too warm and noisy for quantum states to survive. But as we have rigorously established today, warm quantum biology is now a proven reality in enzymes, DNA, and bird retinas. The categorical objection that the brain is too warm has effectively collapsed.
So is that a definitive victory for quantum consciousness?
Not at all. And this distinction is absolutely crucial. The sources provide a very rigorous framework for evaluating claims in this field, breaking the evidence down into 5 distinct grades.
5 grades?
Grade 1 is direct, undeniable biological data, like the kinetic isotope effects proving proton tunneling in enzymes. Grade 2 is biophysical plausibility. Grade 3 is theoretical modeling. Grade 4 is the instrumentation horizon, things we might be able to measure soon with tools like nanodiamonds. And finally, Grade 5 is speculative consciousness relevance.
So we have to be incredibly careful not to conflate the grades. Just because we have Grade 1 rock-solid evidence that quantum tunneling is breaking down toxins in our liver enzymes, that does not mean we automatically have Grade 1. Grade 1 evidence that quantum entanglement in our neurons is creating our subjective sense of self.
Exactly. This raises a paramount philosophical and scientific question. Just because quantum effects can exist in warm biology, does that mean they're the root cause of consciousness?
That's the million-dollar question.
It is. We do have some highly tantalizing clues. For instance, the exact mechanism of how anesthetic drugs erase consciousness remains largely a mystery.
Yeah.
But recent experiments suggest that anesthetics might selectively bind to and disrupt quantum-level actions, specifically within those neuronal microtubules.
And the sources also mention theories that these microtubules could act as something called high-Q quantum electrodynamic cavities. Whoa, hold on. High-Q quantum electrodynamic cavities. You cannot just drop a massive physics phrase like that and expect me to keep up. What does that actually mean in plain English?
My apologies. Let's translate that. Imagine a perfectly mirrored, perfectly sealed acoustic room. If you snap your fingers inside that room, the sound wave bounces off the perfect walls back and forth, echoing almost endlessly without losing any volume or fading away. That is what a high-Q cavity is. It's an environment that perfectly traps and sustains a wave.
Oh, I get it now.
The theory suggests that the physical cylindrical structure of a microtubule in the brain acts perfectly like one of these mirrored rooms, shielding entangled quantum states from the destructive noise of the cell, allowing the quantum wave to bounce back and forth and survive long enough to perform complex computations.
So it's physically plausible that the brain is a quantum computer, but the sources emphasize a critical warning here. Warm quantum biology establishes a precedent, not a free license. It proves that it is physically possible for the brain to use quantum mechanics. It does not prove that it does, or more importantly, that those mechanics are the root cause of our subjective inner experience.
Precisely. To demonstrate that a microtubule sustains a coherent quantum state is ultimately just a claim about structural physics. To claim that the collapse of that quantum state is the subjective feeling of being alive, the feeling of seeing the vibrant color red, or the feeling of being in love, That is a massive, staggering philosophical leap.
It's the hard problem.
The dualism tenet, what philosophers call the hard problem of consciousness, remains completely unresolved. Simply reducing consciousness from complex classical chemistry to complex quantum physics doesn't necessarily explain the subjective inner experience of it. The mechanism of how physical matter creates a mind remains wide open.
So, what does this all mean for you listening to this? Why does exploring this mind-bending quantum realm matter if we haven't definitively solved the mystery of consciousness yet? It matters because it fundamentally and permanently reshapes how we view our own physical existence. Whether or not quantum mechanics explains the human soul, it undeniably, miraculously explains the human body.
It truly does.
Let's recap this incredible journey we've been on. We started with the macro-level thermodynamics of Jeremy England, realizing that your very existence as an ordered, breathing being is a localized defiance of cosmic entropy, driven by the universe's fundamental desire to dissipate energy. We zoomed in to find enzymes performing impossible ghost tricks, tunneling protons through solid energy barriers to keep you alive millisecond by millisecond.
Your ghost is in the machine.
Yeah. We saw how a quantum typo, a single proton momentarily tunneling to the wrong side of your DNA, can permanently rewrite the genetic code, driving the staggering evolution of all life on Earth. We looked at plants perfectly routing sunlight via quantum superposition and birds literally seeing the Earth's magnetic fields through entangled radical pairs. And perhaps most profoundly, we completely reframed the chaos of the biological cell, realizing that the heat and noise aren't destroying life. They are the structured chisel of the decoherence engine, computing our reality out of a blur of quantum possibilities.
It is a stunning, beautiful portrait of biology. But I want to leave with one final, deeply provocative thought that builds upon everything we have discussed today. Currently, human engineers and massive tech corporations are spending billions of dollars trying to build the next generation of quantum computers.
Everyone is racing to build one.
And their entire design philosophy is based on absolute control. They are fighting a desperate war against decoherence, spending vast amounts of energy to freeze out the noise, cool the processors to absolute zero, and force their machines to work in total dead silence.
Because they believe that for quantum computing to work, the environment must be perfectly sterile and frozen.
But look at what billions of years of biology has achieved. If we are finally understanding that life thrives on noise rather than fighting it, that the biological decoherence engine uses the chaotic hurricane of thermal energy to compute with unparalleled, breathtaking efficiency, what does that mean for the future of our own technology? What if the ultimate, most powerful quantum computer of the future isn't a sterile, frozen, multi-billion-dollar laboratory setup isolated from the world? What if the pinnacle of computing technology is warm, wet, organic, and beautifully chaotically noisy? What if the most advanced technology the universe can produce looks exactly like us?
That is a staggering thought to end on. If we can truly master and replicate the biological decoherence engine, we won't just be building better calculators. We'll be building synthetic biology. We will be engineering life itself. Thank you for joining us on this deep dive into the microscopic quantum machinery of your own existence. We invite you to keep exploring, keep questioning the seemingly obvious classical rules of the world around you.
Thanks for watching. Please like and subscribe to Logic Lab 77
quantum ghosts quietly
stay safe.
phasing through walls inside of you, keeping the magnificent engine of your life running.